Hereby, I open this academic ceremony in which Ms. Xiuxiang Tan will defend the academic thesis, Nerve Fibers in the Tumor and Microenvironment, a novel prognostic biomarker in CCA and PDAC. May I invite you to present a summary of your study and the conclusion of your thesis in the next 15 minutes. I give you the word.
Thank you, dear Rector. Good day, esteemed members of the Corona Committee, colleagues, and my friends, here and online. Welcome, and thank you for being here. It's my pleasure to stand here today as a PhD candidate, ready to defend my research. So over the next 15 minutes, I will present my research on the topic of nerve fibers in the tumor microenvironment, a novel prognostic biomarker in cholangiocarcinoma, CCA, and pancreatic cancer, PDAC. My presentation will be in 4 parts. First, I will provide a brief introduction to the research background. After that, I will present an overview of my thesis outline. Then, I will talk about the main results from my thesis. And finally, I will conclude with a summary of my research findings. Let's begin with the first section of my presentation, the research background. As we can see on here, the pancreas is a digestive organ located in the abdomen, while the bile duct is a small tube-like structure formed where the common hepatic duct and the cystic duct join. Cancer is a disease in which body cells grow uncontrollably. And cholangiocarcinoma, CCA, is a cancer that originates in the bile ducts. Intrahepatic cholangiocarcinoma, ICCA, and the perihilar cholangiocarcinoma, PCCA, are 2 subtypes of the CCA, and they have different treatment methods since their locations are different. Pancreatic cancer, PDAC, arises in the pancreas. Both CCA and PDAC are aggressive and neurotropic cancer types with high frequent perineural invasion. Unfortunately, the diagnosis of these cancers often occurred at advanced stages, limiting treatment options. Therefore, ongoing research is essential to identify biomarkers for early detection, prognosis, treatment stratification for personalized care, and development of therapeutic agents. The tumor microenvironment, TME, in which the tumor is located, plays a critical role in tumor growth, viability, and spread. The largest tumor component of the tumor microenvironment is a significant area of investigation, as it's often responsible for poor treatment response. The microenvironment is host to various cell subpopulations, and some of these cells play a dual role in the tumor growth. For instance, immune cells have both stimulatory and inhibitory in the tumor microenvironment. Tregs are a specialized subpopulations of T cells that act to suppress immune response. which is considered a bad role in anti-cancer behavior. So it's a bad cells. While CD8 cytotoxic are protective to the host. So it's a good cell. As I mentioned, PDAC and CCA are considered to be neurotropic cancer with high frequency perineural invasion, which is considered aggressive cancer behavior. So we are considering if there are other role of the nodes in the TME. Since the phenomena of PNI are widely known and investigated, so we are exploring the possibility of other roles of the nerve in the tumor microenvironment, named as nerve fiber density NFD. Nerve fiber density in this thesis is defined as the numbers of nerve fibers with a diameter less than 100 micrometer counted in 20 continuous fields at 200 times magnification. Here, routine H&E staining shows black arrow to the localization of the small nerve fibers, which can show that are here routine H&E staining shows the black arrow to the localization of the small nerve fibers that are not visible on the H&E staining, but can be seen with positive staining in the PGP immunohistochemistry. Of note, these small nerve fibers should be differentiated with pre-existing nerve trunk, which can show tumor infiltration in the epineural, paraneural, and the endoneural space of the nerve, and which traditionally used to define paraneural invasion. The zoom-in image of PGP shows a positive bigger nerve trunk with paraneural invasion, and this big nerve is also easily recognized on the zoom-in routine H&E staining red arrow. Now let's look at the thesis outline together. So the overall aim of my thesis is to study the role of nerve fibers in neurotropic cancer, specifically PDAC and CCA, and explore a novel prognostic biomarker. This thesis consists of clinical observational studies that focus on the function of nerve fibers in PDAC, PCCA, and ICCA patients undergoing surgery. Before we start our researches, we provide a review of current knowledge of nerve fibers participating in the progress of neurotropic cancer and summarize retrospective results of previous literature about nerve fibers in PDAC and CCA patients. In Chapter 3, we aim to define role of small nerve fibers in the TME and their spatial arrangement to immune cells within the TME of PDAC. In Chapter 4, we explore nerve fiber density in correlation with the survival of PCCA patients. In Chapter 5, we demonstrate nerve fibers as a novel and important predictive biomarkers in ICCA patients. Checkpoint inhibitors have based immunotherapies that target PD-1 or CDLA4 pathways have achieved impressive success in treatment of different cancer types. For example, antibodies targeting PD-1, PD-L1 axis have been approved as second-line or first-line therapies for an ever-growing list of malignancies. including melanoma, lymphoma, lung cancer, gastroesophageal, and renal cell cancers. However, the majority of CCA patients receiving checkpoint inhibitors do not derive benefits. We urgently need new biomarkers to predict response to checkpoint inhibitors, which can help patient selection. In Chapter 6, we identify NFD to correlate with PD-1 positive T cells as a biomarker for a good prognosis. In this section, let's talk about the main results of this thesis. So our findings indicate that high NFD is associated with a better survival in PDAC, ICCA, and PCCA patients undergoing surgery. For example, in PDAC, patients with high NFD show a median 5-year overall survival of 34 months, while patients with low NFD had a median overall survival of 22 months. Further, we performed immunofluorescence and found that nerve fibers in the tumor microenvironment are co-localized with lymphoid aggregates, and the lymphoid aggregates, which are co-located with nerve fibers, consist of malign B cells, here shown in yellow. We applied immunohistochemistry on perineural invasion and nerve fiber density slice, respectively, to explore where these small nerve fibers originate from. We have shown that they are specifically from parasympathetic origin nervous system, the purple innervation on the figure. PGP is a general neuronal marker. NPY is a marker for sympathetic origin. And the VIP is a marker for parasympathetic origin. On perineural invasion slide, you can see PGP staining yellow arrow being expressed demonstrating cancer invasion red arrow. The sympathetic origin staining is positive. The parasympathetic origin staining is negative. On nerve fiber density slides, you can see small nerve fibers stand positive in the PGP immunohistochemistry, negative in the sympathetic origin immunohistochemistry, and the yellow arrow points to the positive staining in the parasympathetic origin immunohistochemistry. We applied multiplex immunofluorescence and method which provide a practical workflow for the simultaneous detection of 8 biomarkers plus nuclear content stain within an image. So we applied 2 panels, 16 antibodies total. Our results show in high NFD group, the CD8 PD-1 positive and the CD8 PD-1 PDL2 positive expressed higher than that expression in low NFD group. From the above, to summary, we've covered many ground so far, but before we wrap up, there's one more section to go. So in this section, I will summarize our findings and draw some new final insights. To summarize this thesis, what we know is 1. High NFD is associated with a better survival in PDAC, PCCA, and ICCA patients undergoing surgery. 2. Nerve fibers are co-localized with lymphoid aggregates, which consist of malign B cells. 3. The parasympathetic nerve fibers are indicative of better survival. For PD-1 CD8, cells are associated with the high nerve fiber density in PCA patients. Based on the clinical observation and the lab results from this thesis, we conclude that nerve fibers can be a potential biomarker to improve clinical outcome by a rational development of new therapy strategies. Though there is still more studies done to confirm these positive functions of nerve fibers in cancers, we expect that these results would provide reference for further studies. It will be broadly applicable to other malignancies. That brings me to the end of my presentation. Thank you for listening and attending. Thank you again.
Thank you very much, dear candidate, Ms. Tan, for your clear presentation. It was well drawn, and I think your last conclusions were well made. And we will now start to have some discussions about your thesis. And I would like to start and give the first word to open the opposition to Professor van Engeland. She was also the chair of your assessment committee, and also virtually she is today my secretary. She is involved as head of the Department of Pathobiology of Cancer, and particularly the role of epigenetics at the Department of Pathology at our university, and I'd like to give her the first word.
Thank you, Mr. Pro-Rector. Dear candidate, congratulations with this beautiful thesis. It was a pleasure to read your thesis, and I would like to express my congratulations to your supervisors. But we are here to ask you some questions about this thesis. And I would like to start with discussing a little bit about biomarkers. What is a biomarker? When do we call it a biomarker? And when can we translate biomarkers to the clinic? So I'm working on biomarkers myself, cancer biomarkers. And in my experience, promising biomarkers hardly make it into clinical practice. You showed some very convincing results for the fact that high nerve fiber density is associated with good prognosis in pancreatic and cancer, but also in cholangiocarcinomas. What is your expectation of this biomarker? Do you think it can be translated to the clinic? And if yes, do you think the biomarker is ready for translation to the clinic? Or if not, what should we do to bring it to the clinic?
Okay, thank you. Highly esteemed opponents, thank you for your kind words. And this completely many questions. Let me answer from the first. What is the biomarkers? So the biomarker, the exact define of biomarkers is like a goal in a many cells, and you find a key, then target that key. You will improve the patient treatment. And for the question, what can we translate the biomarker to clinical application? So in our study, we find nerve fibers as a prognostic potential prognostic biomarker in cholangiocarcinoma and pancreatic cancer. Though they are just primary results from our studies, and the study samples is not enough, so it's not very big. But from our we have done 3 or 5 studies for investigating the nerve fibers, and we all show a positive function of the nerve fibers in PDAC and CCA patients. And now we were able to show this is a positive biomarker. In the future, we hope we can explore the potential mechanisms of how these biomarkers done, and develop a new drug to stimulate the biomarkers to stimulate the nerve fibers grow, and combine with the immunotherapies and the nerve fiber therapy. Since the innovation cancer treatment will emerge from the combinatory drug strategies by targeting not only the cancer cells but for the stromal components. So, and the nerve fibers also a part of the stroma of the TME. So, there is still a long way to apply the biomarker to the clinical, but we hope in the future we can find a drug to target the nerve fiber and stimulate the nerve fiber growth, then combine with this with the immunotherapy. Thank you. I hope this can answer your question.
Yeah, partly it did, but I would like to point to your conclusion, which I completely agree with, that you showed the data in one study, one population, one cohort, right? One patient cohort.
We show the data in ICCA cohort, PDAC cohort.
Yeah.
Yeah.
And the results all show in the same direction, right?
Yeah.
So I think that's very strong evidence that you see this effect in 3 different tumor types.
Yeah.
But in our experience, it's also important to validate these findings in another lab. So a completely independent study that confirms your data. So validation is very important because that underscores the strength of the biomarker. Are there any plans to collaborate with other labs or to see if these data can be validated independently?
Dear highly esteemed opponent, so can you give me an example of to classify your question?
I give you an answer, but we cannot continue the condition, so I have to interrupt. I'm sorry.
Okay.
Yes.
Well, thank you very much for your response, Xie, and I'll give the word back to the program.
Thank you.
Thank you very much, and the opposition will be swiftly continued by Professor Dahl. Professor Dahl is involved in the Department of Molecular Tumor Biology in the Department of Pathology at the RWTH Aachen, and he was also a member of your assessment committee, and I'd like to give him the word.
Yeah, I would also like to congratulate you for this very fascinating work and your great dissertation. I think you had 6 publications out of the work and 1 first author paper in oncogene, so that's a great achievement. And also the cover of your booklet is very intuitive because it shows the close, intimate connection of all the cells interacting in the tumor microenvironment. And your, what's the name? Paranyms colleague, they presented. Thank you for that. So I start with the first question. Manon van Engeland already talked about biomarkers, and now you have this finding that a high nerve fiber density of the small nerve fibers is connected with good prognosis, but some tumors don't have it and others have. If, of course, we would like to go for the pathways involved and also some genes which are responsible for this phenotype. And how could we address this experimentally? I can give you a little clue. We have started in a project together with you where you have been involved where we compared some different types of cholangiocarcinoma. Can you summarize that a bit?
Dear highly esteemed opponent, thank you for your kind words. So your question is about the mechanism, the methylation, the gene. Yes, I also participate the study our cooperation with the HOPIC gene. The HOPIC homeo gene is a transcription factor expression and is epigenetically silenced in many malignancies, such as gastric cancer, lung cancer, and colorectal cancer. I hypothesize because for our cooperation, the project, the DNA isolation, the quality of DNA isolation most very low. The quality is very poor, so we don't get a very positive function results.
Yeah, maybe for the audience it would be to go one step before that. You mentioned a gene which we have identified to be differential between high and low nerve fiber containing cholangiocarcinoma. But how did we achieve that? What did we compare? So how do you experimentally proceed to come to such candidate genes that may be involved to constitute this phenotype?
Okay, so in the proposed experiments, I think we can use the wide genome-wide methylation error experiments to detect the methylation level of HOPIC gene, to check the different group of high NFD and the low NFD group, and to check the level of the HOPIC gene methylation.
Thank you. That's what I wanted to hear, that you make two cohorts, one with a high nerve fiber density and the other with a low.
And compare the HOPIC gene methylation level.
Exactly, and then we compared some level of methylation. So what could you, that's already my final question, what do you know about the meaning or the importance of epigenetics in cancer, especially DNA methylation? Why is it so important?
In my knowledge, the transcript factor expression is often epigenetically silenced in many malignancies. So the latter gene is in relationship to the cancer in participating in cancer progress, like lung cancer, gastric cancer, and colorectal cancer. And the high methylation of the gene in many malignancies, the transcription factor is often silenced when cancer happens.
Yeah, but maybe just one final word about tumor suppressor genes in principle. How can they be inactivated?
How can they can be activated?
Inactivated tumor suppressor genes, two ways in the cancer, how they get, so to say, killed, dead. That's no more functional. One is epigenetic, you said, for instance, DNA methylation, and the other would be?
High methylation is relation to aggressive tumor behavior, and the cancer, the malignancies often show the gene is silenced. So the treatment stratification is to reactive the silenced gene. It is the direction of develop the treatment strategies.
Yeah, that's a possible way to reactivate it. Thank you.
Thank you very much. Now the opposition will be continued by Professor Lang. He is involved in the Department of Liver Surgery at the RWTH Aachen, and I'd like to give him the word.
Thank you for the introduction. Dear candidate, congratulations for this very nice work and this very nice presentation. I just have a few questions. First, you were focusing on pancreatic cancer and cholangiocarcinomas in your work. Is there anything known about the meaning of neurofibril density in other tumor entities, particularly in gastrointestinal cancer?
Yes. Dear highly esteemed opponent, thank you for your kind words. And your question is there are other researches of the investigated the neurofibril density in other entities carcinomas. Yes. The neurofibril density is widely investigated in gastrointestinal tract cancers like gastric cancer, colorectal cancer. But the neurofibril density, the roles neurofibrils play is quite different in different types of cancer. For example, in gastric cancer, researchers find the proper innovation is critical at the edge of the tumor heterogeneous. So that's a negative function of the neurofibrils in gastric cancer, and also in colorectal cancer. The more neurofibrils are indicated poor clinical outcome. In my knowledge, I think I do believe the biology of different organ and different cancers are quite complete different. For our PDAC and CCA, they often show large stroma in the tumor microenvironment, while for gastric or colorectal cancer, they grow in a solid pattern, so they don't have so much neurofibril density and so much stroma. So when we talk about the neurofibrils, because it's a part of the stroma members. So when we talk about that, we need the large stroma. Yes, this is my answer.
Thank you. Another question is the assessment of nerve fiber density. Is it possible to determine this by frozen sections?
Frozen section? Dear highly esteemed opponent, thank you for your question. Frozen section often demonstrate perineal invasion, and this can be a reason for R1 resection. PNI often show away distance from the main tumor bulk, and somehow the cancer cells pre to spread through the nerves, and they use the perineal space as a way of least resistance. The frozen section we are not very good for count neurofibril density. Since the neurofibril density is very small and we can see only H&E staining, we need the special staining to recognize the neurofibrils. So it's not very good method to recognize on frozen section recognize the nerve fibers on frozen section.
Thank you. One more question. If there is no role for nerve fiber density in the change of a surgical procedure, so what do you think is the clinical relevance for the future?
So in frozen section, it's not very important to know the nerve fiber density, since the high nerve fiber density can't change the surgical procedure. But at present, we were able to show the nerve fibers as a strong prognostic biomarker to predict a better clinical outcome. And in the future, we hope we can develop a new therapies that can target nerve fibers and stimulate the nerve fibers grow, then apply that in the cancer treatment, maybe combined with the immunotherapies. Since the innovation cancer treatment will emerge from the combinatory drug strategies targeting not only the cancer cells, but also for stromal cells. Thank you.
Thank you.
Thank you very much. Then the opposition will be continued by Dr. Groot-Hoelkamp from Rotterdam. He's affiliated with the Department of Surgery from the Erasmus Medical Center in Rotterdam, and I'd like to welcome him and thank him that he joins us this afternoon. I give you the word.
Thank you, Mr. Prorector. Dear candidate, I think you have accomplished, I want to congratulate you with an impressive thesis, and also congratulate your thesis team. I was particularly impressed not only with the content, but also with the artwork. And I just heard that you made all the drawings yourself. And I think this is the most cheerful thesis cover I've ever seen.
Thank you.
So it's really impressive. I have a few questions for you. And my first question is about chapter 5. In chapter 5, you look at patients who had a resection for intrahepatic cholangiocarcinoma, and you look at nerve fiber density and to see whether it predicts survival. And if you look at figure 3 panel E, what I think is very interesting for me as a surgeon is that you found a subgroup of patients based on nerve fiber density and a node-positive disease that have a very poor prognosis. That's the same as systemic treatment. So in hindsight, it would be better not to operate those patients. But what should I do as a surgeon to avoid operating these patients? What can we do? How can we apply this knowledge that you gained in your research? How can I apply that for my next patient with intrahepatic cholangiocarcinoma?
Dear highly esteemed opponent, thank you for your kind words for my thesis. So your question is, how can we include more patients that were excluded in this cohort, in this study?
Yes. So how can you show that if nerve fiber density is low and if a patient has node-positive disease, that they die rapidly? And so the surgery is very risky. It's big surgery. So it's better not to do it, but we didn't know in advance. So could we do, for example, a biopsy before the surgery? Usually we don't do a biopsy for these tumors, but maybe should we do a biopsy so you can tell me what the nerve fiber density is?
Okay, I see. So again, let me explain how we can count the nerve fiber density in our study. So the nerve fiber density, we need the H&E staining and the immunohistochemistry stain the nerve fibers. When we count them, we need they are counted in 20 continuous fields at 200 times magnification. But for biopsy, it's too small, so we don't have enough fields to count the nerve fibers. So at present, we can't use the biopsy to investigate nerve fibers. So and actually, this is also a limitation of our methods to define the nerve fiber density. And this is also a direction of our further study to explore more methods to counting the nerve fibers and to define the nerve fiber density. Then we can utilize the spicy. Then we can utilize more samples, include more patients.
Okay, thank you. I have another question regarding how we can use this knowledge in the future for patients. So, do you already know whether the nerve fiber density are just like an innocent bystander, like a reflection of a favorable prognosis? Or do you believe it is really in the causal pathway, so that these nerve fibers themselves are really doing something good, something favorable, and that we should develop treatments that increase nerve fiber density?
Dear highly esteemed opponent, the small nerve fibers we find is a prognostic for a better survival. Is we find that is originate from the parasympathetic orange. So we don't think the sympathetic orange nerve fibers will prognostic a better clinical outcome. So it's not all nerve fibers are indicative of good prognosis. So we have the specialized nerve fiber orange for a better.
Okay, thank you. I'm asking whether I have time for another question.
There's even time for 2 questions if they are short.
I have one more question. So maybe you will continue doing research, and maybe you'll continue doing research with nerve fibers. So imagine you get a big grant from the Chinese government, 1 million euro. It's probably not euros, but I don't know the currency exchange rate. And you'll have to decide what will you do with it. Will you use it to better understand the nerve fibers, so really pathophysiological and etiological research? Or would you get in touch with clinicians and get larger cohorts and really see how you can use this biomarker to improve patient care? Is it ready already for the clinic, or do we need more basic science?
In one hand, we need more patients to confirm, and in other hand, it's also very urgent to investigate the exact molecular and cellular mechanisms about the nerve fibers. How to work, why the nerve fibers predicted, what the interaction between the nerve fibers and the tumor cells. So if I have 1 million grants, based on my biological major, I would prefer to choose the molecular and the cellular mechanism exploring.
Good. I'm glad you made a decision, because it's very easy to say you want to do both, but that's very clear. Just to conclude, I want to say that I'm very impressed that you're doing all this in English. My daughters are learning Chinese in school, and they're not ready for public speaking in Chinese. So I'm very impressed. You're doing a good job. Congratulations.
Thank you.
I'd like to give the word back to the rector.
Thank you very much. Then the opposition will be continued by Dr. Bouwense. He is affiliated with the Department of Surgery at our Maastricht University Medical Center Plus. Yes, and I'd like to give him the word.
Thank you, Mr. Pro-Rector, dear candidate. Congratulations with this great work, and of course with the artwork. It really helped me out. I'm a surgeon. I'm not a pathologist or a molecular biologist. So it was a bit of a refresher for me for my medical education. So thank you for the great artwork. Congratulations also for your team, for your promoter and co-promoter. And I want to discuss with you the clinical application of the nerve fiber density, what you found. And we already discussed the role of the biopsies. And I was wondering if you have any results from your research or from the literature on how we can use the nerve fiber density and the small nerve fibers to select patients. Because as you know, we not only operate on patients, for instance, for patients with pancreatic cancer, we try to select the good patients and try them to treat them nowadays neoadjuvantly with chemotherapy. So how can we use your technique to find the good patients that perhaps respond on chemotherapy or on immunotherapy?
Dear esteemed opponent, thank you for your kind words. So refer to the biopsy sample. Again, this is a limitation of our methods. We at present we can utilize the biopsy to detect nerve fibers. But our method in our study we include the patient criteria. The first one is the patient who are able to who are eligible for surgical treatments. Then we can get the whole slice imaging. Then we can count their nerve fiber density status based on the whole imaging slice. So at present, the biopsy samples we cannot utilize. But in the future, we will develop, we will explore better methods to define the nerve fibers on biopsy sample. Then that we can include more patients in our study. Thank you.
Thank you. And regarding the patients, now you got evaluation of the tissue of patients that were all operated on. And did you see within your cohort differences between patients that were treated with chemotherapy and the patients that weren't treated with chemotherapy or with other experimental systemic therapy? So do you think that there is a place for nerve fiber density to select patients, for instance, for immunotherapy or for chemotherapy? And if not, how would you do that?
Thank you, esteemed highly opponent. Regarding this question, my answer is yeah. We can use the nerve fiber density to select a patient for stratification treatment therapies, because in our study we found nerve fibers, the potential mechanism of nerve fibers may be based on the communication between nerve fibers and the immunotherapies. So nerve fibers play an important role in the crosstalk between between other components in the of the stroma, and the nerve fibers are co-localized with with the clusters of immune cells. So if when we check the status of nerve fibers, we can maybe this patient is more suitable for the immunotherapy, because in our hypothesis that the nerve fibers co-localization with nerve fibers maybe improve the immune response. Yeah.
Thank you. Is there space for another question? It would be great to have this technique, of course, suitable for biopsies to better predict immunotherapy, perhaps a success in those patients. And you showed that if there are more small nerve fibers in the tissue, it's beneficial, and also when the immune active cells are there. What are ways to promote nerve growth in that tissue and get the good cells over there? What do you think?
Dear highly esteemed opponent, in my opinion, the nerve fibers attract immune therapies. So nerve fibers include the immune cells, which can improve, which maybe increase the immune response, that lead to a better, that may be suitable for immunotherapy. So I think the mechanism of this is the nerve fibers attract immunotherapies, including around the nerve fibers that increase the immune response.
Okay, thank you. I have no more questions. Because we're back to you, Mr. Prorector.
Thank you very much. Then the opposition will be continued by Dr. Severance Rijvers. She's affiliated with the Department of Pathology and is working basically in the field of cancer pathology at the Maastricht University Medical Center Plus. And I'd like to give her the word.
Thank you, Mr. Prorector. So, dear candidate, first of all, I would like to congratulate you and your team with your hard work for this thesis. I really enjoyed reading it, and as a pathologist myself, I really like this simple immunohistochemical staining as a biomarker. I was also very impressed by your artwork and your cheerful cover, and it put a smile on my face every time I opened it. So my first question, I would like to take you to the propositions accompanying your thesis, and I would like to ask one of your paranames to read proposition number 6 out loud.
Basic research is what I'm doing when I don't know what I'm doing.
Yes, thank you. So I was wondering, how does this proposition apply to you, and have you figured out what you are doing yet?
Dear highly esteemed opponent, thank you for your kind words. When I conduct this thesis, in my opinion, the basic research is a process that is a process of discovering, including exploring the unknown and testing the hypotheses, even though the outcome is uncertain. In context of my research and so the cancer research, we are constantly explore new methods and validate the new ideas in search for a better understanding of cancer. This often involves conducting experiments and studies, even though they may have not a very obviously or immediate application in the clinical, but that contributes to our overall understanding of this disease and its underlying mechanism. For example, in my research, we done with the stroma, we explore the tumor microenvironment, the stroma components. So we contribute to the development of certain types cancer may contract to identify potential therapeutic strategies to better understanding how these components work, and this research may not have a direct application or immediate application to the clinical outcome. But I hope in the future we can develop the new therapy, the new treatment therapies that improve cancer patients' life and improve our understanding of this disease. So exploring unknown. So let us say I don't know what I'm doing, but it allows us to explore the unknown and make a new discovery that can eventually lead to improved outcomes for cancer treatments. Thank you.
Thank you. So I further noticed when reading your thesis that you use different cutoff values for your nerve fiber density in the different tumors you investigated. So in chapter 3, for example, in the pancreatic duct carcinoma, you used the same categories as have been described in breast carcinoma, which are negative or weak expression or moderate to strong expression. But for your studies on cholangiocarcinoma, you used ROC curves to determine the cutoff values, but these were also different between the different types of tumors. In perihilar cholangiocarcinoma, the cutoff value was less than 10 versus 10 or more fibers, whereas in intrahepatic cholangiocarcinoma, your cutoff was 0 fibers versus 1 or more nerve fibers. So these are quite different. And I was wondering if you could explain these differences in cutoff values in a pathophysiological manner.
Thank you for your question. So your question is to explain the different cancer types of ICCA, PCCA, and the PDAC.
Yeah, but why do they have different cutoff values for your nerve fiber density?
Yes. When we count the nerve fiber density in these 3 different types of cancer, PDAC is more heterogeneous, and the nerve fibers often show in the center, and in the center, and the nerve fibers are very frequently show in the tumor bark, but in ICCA, the nerve fibers are more show in the edge of the tumor. Let's we find the different feature of the nerve fibers in these 2 types of cancer.
Thank you. Okay, so another question I have is whether you considered, investigated blood vessel distribution in your slides, because nerves often travel together with blood vessels and they share the same distribution. So I was wondering, did you ever consider it, or is it, are you planning to study this further or not?
Dear highly esteemed opponent, regarding the blood vessel, to investigate nerve fibers in blood vessel, in my opinion, the blood vessel also like the feature like the nerve fibers. So nerve fibers.
Dear Ms. Candidate, you are allowed to finish your answer, but you're also allowed just to finish. To stop.
We can talk later.
You can talk later. Okay, then we finish now, and that basically is we have to continue as follows. Dear Ms. Tang, dear Ms. Candidate, the time appointed for defending your thesis has now passed. The degree committee here present, we will withdraw from this audience, and we're going to discuss the quality of your thesis, and in particular the way you have defended your thesis this afternoon. I request that you and your colleagues over here wait for our results, for our deliberations, and our return to this aula. Thank you very much.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No no place like home. If it's questions that you've got, go the extra mile or die. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Dear Ms. Tang, dear Ms. Tang, dear Ms. Candidate, the degree committee here present has assessed the quality of your thesis, but in particular the way you have defended your thesis this afternoon. In view of its positive verdict and taking into account your previous qualifications, the degree committee here present has decided to grant you officially the degree of doctor. Professor Neumann is authorized to confer upon you this academic distinction in accordance with Dutch University customs, and I invite your first promoter to now take the floor.
All right, please.
Do you promise to work in accordance with the principles of scientific integrity or at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
That's good. But also you vested in us by law and conformity with the decision of the committee hereby present. I hereby confer upon you, Xiuxiang Tan, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the director, the secretary, and the other members of the committee, and affixed with the official seal of the university. You can conform with your camera. Congratulations.
I give the word now to Dr. Hay, who will speak the laudatio to the candidate.
Thank you. Esteemed Dr. Xiuxiang Tan, dear Xiang, you have done it, Gongqi. I hope I just said congratulations in Chinese, otherwise I apologize straight away. I'm honored to congratulate you with this doctor title. Also on behalf of Professor Neumann. This is a meaning of endurance and resilience you have shown during the last years while working in my lab. I also wanted to take the time and congratulate your parents and your family members. I know they supported you online during the defense today. Thank you to the people here in the audience who made it out here. Dear Xiang, we can only imagine how big the culture difference is between China, the Netherlands, and Germany. I'm sure I would not have survived for 1 second without help in China. You have been so brave. You did so well in overstepping fears, and you just embraced the experience. Approximately 4 years ago, we were teamed up by coincidence. You were ready to change your research team, and I had asked for extra hands to help out with my research. I was unexperienced, and we really started our projects together. I treasure the days we were able to spend time together and work on developing new technologies and share research ideas. On the other hand, I also felt bad for you because my work was so unfocused, and you never told me you didn't have time. I will give you and the audience one example. The first project you started on was annotating pancreatic cancer histology slides in a very detailed way. I thought it would make sense to define tumor region and stroma region very precisely, and this potentially would correlate with survival. You are not trained as a medical doctor, but you learned quickly. We spent days sitting together and I explained you in detail which cells were cancer cells and which ones were benign, something that can be hard for a specialized pathologist as well. By doing this, you gained so much pathology experience in recognizing tumor and the tumor microenvironment. But it was also very labor intensive. It took you 1 slide per day, and we had over 200 patients in our cohort. In total, you were occupied with this for months, and you know what? Nothing came out. My hypothesis was wrong. I felt so bad for myself, but also for you. I felt responsible to make sure you would go home with a finished PhD. You started writing a review and focused on nerve fibers, and this worked. You did this very independently. You read over 100 papers, and this was accepted by a very good journal. Your manuscript has gained many citations by now. In the meantime, I had been thinking about the pancreatic cancer slides. Still being frustrated, nothing came out. We started counting small nerve fibers, and now the topic of your thesis was born. During the 4 years you were here, you used your opportunities to the max. You followed me to the University Hospital of Aachen in Germany. Here you integrated into a new group of scientists, and you did well. I was happy to have a friend who was just as surprised about cultural exchange as I was, and we laughed about our issues. You followed German and English classes. You learned new techniques, such as AI script writing and developing deep learning research plans. You are a co-author on some very high-ranking manuscripts, coordinated by the group of Jacob Kater. Your own deep learning work was selected as a poster at the German Pathology Days in Münster. Your first authorship manuscript is under evaluation in a good journal. Also in the wet lab, you learned many new techniques. My friend and good colleague Shivan Sivakumar at Oxford University shared how to make single-cell suspensions from pancreatic cancer tissue. We focused on cholangiocarcinoma tissue and adjusted the protocol. We chased blood and tissue samples throughout the hospital. You always knew where to find samples and how to get into areas where we didn't have access to. Here you showed you are able to push through and not give up. When I was thinking about what to write for you, I checked my iPhone for help. My iPhone photo library recognizes my children, the cat, and you. I have so many pictures on my iPhone from you and our research. You always texted me when you had a problem and when things were running smoothly, usually with a picture. I have the pictures printed and you will find them in your present. You are a very responsible person and you can be trusted with the lab work being taken care of. On a personal level, it has been an enrichment to spend time with you. We had time to get to know each other while sitting and driving in my car for hours, looking for deer in the woods between Maastricht and Aachen. I was so concerned when COVID started and you were in China, when the lockdown started. I was happy you got to see your family, but even happier when you made it back to Maastricht. I was afraid flights would not be available. Then a long time started with home office. We were not able to meet and you worked from home. This was not easy and a very isolated time for you. I remember driving around in the evening in Aachen trying to find you when you visited the famous Christmas market. I left you alone with the other researchers because I needed to finish some diagnostic liver biopsies. When I was ready to pick you up, we were confused where to find each other. I was happy after driving for quite a while I had you back. I was afraid you were lost in the dark in Aachen. We did groceries together in the Chinese supermarket, and also my kids love you. You made dumplings for us and you integrated with some of my au pairs, since you both needed to learn English. While sitting in the car for hours, we always looked at the black and white cows. They are everywhere and outside in the fields for many months of the year. You were amazed when I told you the cows can be hugged and that they're very friendly animals. I knew it was your dream to go cow hugging one day, and I promised you we would do this together. Last year, we made it to a cow hugging farm, and we spent hours with the cows, experiencing their body warmth and their kind personality. Cow wellness is a Dutch tradition and it's good for the brain. It slows you down just to sit with them and think about life. Mental health matters. Remember this when you're back in China. I know you have a very polite personality, and sometimes you can even be shy. So I thought, let me use this time to you to point out some of your strengths. It's so easy for me to name them, memorize them well, because you will have upcoming job interviews. You are able to adapt to almost any situation, and this is not something anyone can do. New cultures, language obstacles, integrating in new groups, you are able to adapt. Don't underestimate this. It might take you a bit of time, but you have proven to be successful. Believe in yourself, stay strong, and just do not give up. You're outstanding in graphical design. You've made fantastic figures and PowerPoint presentations, as we have seen today as well. You are able to catch the main message quickly, and you can translate this into creative figures. This is a skill that is exceptional and very much needed in every research group. I will for sure miss this. Most of all, thank you for your loyalty, Xiang. I've always felt happy to work with you, and it was a pleasure to have your support. The experience of having you in my team made me think about my own personality. I'm afraid I have not brought the same joy to my bosses. You taught me to be more patient, to be loyal and less demanding, and while I'm still improving this concept, I will have you in the back of my mind. I'm sure you will have a great future ahead of you as a postdoc in HPV cancer. I hope we keep on working together, and I wish you a safe journey back. Congratulations once again to you and your family. We are very proud. Thank you so much. I will now give the word back to the Pro-Rector.
Thank you very much. And I would like to start to congratulate the new young doctor. So, Dr. Tan, welcome.
Thank you.
And this is in the Netherlands and in Europe, the highest degree which you can receive. And I would like to congratulate you with this degree. You did a splendid job, and you did a splendid defense. In my congratulations, of course, I also bring forward the promotion team. They put a lot of effort for you in the last years to bring you to this afternoon in which you defended your thesis. And I also would like to thank the members of the Corona who were here today. They only had 7 minutes, but I think it takes more than 7 minutes to prepare for the questions to be addressed here and to be here. In my congratulations, on behalf of Maastricht University, of course, I always look a little bit in the camera and to the people who are now online in China, who are your family, your friends in China. I know you will, I received already a notice that from CSC The Hague, that you will return back to China to do your 2 years of voluntary work in China, and you go to Fudan University to work over there, most likely. Keep in mind that you have for Maastricht University a very important role. You will be now an ambassador of us. You will be an ambassador from Maastricht in China, and we're going to use these ambassadors in the near future to start up exchange programs. So it might be in Fudan that there will be Dutch students who will join your team if you start a team over there. That's an important thing. And we have a word for that. We call it now officially science diplomacy. That's what the ambassadors are going to do. You did a great job. I think today is a day which I learned when I was in China myself. It's a day for you. You're going to shine today, and you're going to relax and enjoy the day. And this, what we're going to do is now to enjoy. We're going to have a little celebration afterwards. And before we go there, I'm going to officially close this ceremony. And for this, I use my hammer, and I close this ceremony. How are we going to continue? Well, we're going to make a picture here as the whole group on the stairs of the hall here in the aula.